Thank you. So I'll try to keep it non-scientific as best as I can. I don't want to show you lots of formulas and experiments and things. To start with, this is not just my license plate, it's our mission statement in my lab. So I'm not going to retire until I find a cure for at least one type of cancer. If that means I'll work until I die, fine, I'll work until I die. No retirement. We have this term that we use a lot called the value of death. So this person here represents me and my colleagues, scientists. I'm not a physician. I finished medical school but never practiced medicine. So I've been working in the lab for my entire life, and we try to find new drugs for treating different types of cancer. This side of the valley on the other hill represents our clinician colleagues. They are treating the patients. So we have lots of good ideas in the lab, but there is really a big gap here to translate those ideas to new treatments that can be given to patients. Believe it or not, it's really easy to kill a cancer cell in the lab in a Petri dish. You can pour water on it. If you put enough water on a cancer cell, it will die. But if you give enough water into a human to kill the cancer cell, you will kill that person too. Or if you put bleach on cancer cells, it will die. You know, our former president had that idea about COVID. But again, the outcome is obvious. If you give that much bleach to a person, you will kill the patient along with the cancer. So we have lots of experiments, like Dr. Crumpton said, but there are lots of drugs that work great in the lab when you test with cancer cells that we take from patients in growing culture, or when we put those cancer cells into animals and test them, it worked great. But even those, they work great in the laboratory. When they go to a patient, many of them do not work. There are lots of different reasons for that, but that's why there is still this gap and value of that we just cannot translate everything we have done in a research lab for a true and useful treatment for patients. I'll tell you about some of the experiences we had when we were trying to develop new drugs from a research lab going into the clinic. So this is sort of our general approach. The big problem in our fight with cancer is that cancer cells are our own cells. They're not like something coming from outside and invading our bodies. It's not like the COVID, which is a virus, which is a foreign organism coming into us. So our immune cells can see them as foreign and fight them. It's like fighting a war with a different country. There are different soldiers, they have different uniforms. You know who the enemy is, you know who is your friend, and your soldiers or police force can differentiate them and kill them. Cancer is like fighting a domestic terrorism. The terrorists are human beings, they're people like any other citizen. Your police force has no way of looking at a person in their face and picking up, is this person a terrorist or a normal civilian? Sometimes you may get a tip saying that, oh, terrorists are wearing blue jackets. You can, send your police force and kill all the people walking on the street with blue jackets, but now you're killing lots of innocent people too. And they learn and they take the jacket off, they put on a green jacket. Or sometimes we take really drastic measures. When you do an amputation, it's like putting a nuclear bomb in one of your own cities. So you're just killing lots of innocent bystanders. So idea is, and even then, some of those are getting on a train, on a plane, and they go to a different city and colonize again. Now that's what you have in metastasis. So what we are trying to do, we're trying to find these small differences, even though these cancer cells, like human normal cells, because that's where they originated from. If we can find those differences, and can we develop drugs to specifically target those differences? So for different types of bone tumors, we always start with one of those differences. We look at building blocks of a cell. These are called proteins. And we're trying to make a small drug that will bind to this protein. We have technologies that we can do to screen through thousands of small molecules to find which one directly binds to this target. And then once we find the binders, we take it another step and try to test those binders to see, do they just bind, but do they also bind and inhibit the function of this protein? And if it does, eventually we want to test this in a whole organism. So we put these human cancer cells into zebrafish, which is really a useful method, or mice, and we try to cure the cancer that we created in these small animals by the drugs that we discovered because they're binding directly to our target. If it all works, of course, we would like to, or we will have to test them in human clinical trials because everything worked this far may still fail when it comes to clinical trial. And one of the problems there is, like Dr. Crumpton mentioned, you cannot do this in an academic environment. You have to work with pharmaceutical industries sooner or later. So we have done this approach for a couple of different targets and diseases. For Ewing sarcoma, we targeted this protein called EWS-FLI, and we found an inhibitor. So that's as far as we could do in the lab. We can do all the experiments we can do with cells and animal models, but that's where we're done. So at that time, the drug companies come to you, you talk to them, they like the idea, they like the drug, and then somewhere along that conversation, they ask, how many patients are there? So when we tell them there are about 400 cases in the United States a year, that's the end of the conversation. Have a nice day, they leave. Nobody wants to make a drug for just 400 patients. That's the horrible truth about the business of the drug development. We were lucky enough that for this drug, I had a grant from Department of Defense, and I was able to show that the same drug may also work for prostate cancer. Now that you have 100,000 patients, and drug companies were interested, they licensed the drug from the university. A small company is running the clinical trial with this drug in phase 2. They just stopped it. We can talk why. But that was the path of a drug that started in our lab and made it all the way to patients. And that path took us 16 to 18 years. If we had enough funding, it should have been done in 3 to 5. But with the rare diseases, rare pediatric sarcomas, the funding is limited as well, so it takes that much longer. We had really good success with another project in the lab for osteosarcoma. We made a drug for a protein called Üren, and it works great in everything we do in the lab. But again, economic reality, for this compound, the one that we discovered, it's working best, it is a known compound, so nobody can have a patent on it. And if you cannot have a patent on a compound, drug companies do not want to invest running clinical trials with this because they can run the clinical trial, then anybody can make and sell this drug. So we are trying to find government funding or private funding to advance this thing into clinic. One opportunity here is that with osteosarcoma, again, the number of patients are in the United States very similar to Ewing sarcoma. It's relatively rare, but osteosarcoma is relatively common in pet dogs in our homes. We have about 400 to 500 osteosarcoma cases, but 10 to 15,000 dogs get osteosarcoma in the United States, and there are lots of pet owners that are willing to participate in clinical trials. So we're trying to start a clinical trial on pet dogs who get spontaneously osteosarcoma and see if this drug works on dogs. If it does, it will be an easier bar for us to push for a clinical trial in children. But that too is a very expensive process. Running a clinical trial in pet dogs would cost us about $1.5 million in the US. I keep writing grants to get this thing, but since it's just a clinical trial in dogs, NIH doesn't really support that kind of research. I would like to finish in 1 minute by telling you the project that Bone Cancer Research Trust supported for Ewing sarcoma. In this case, this is like a drug repurposing project. When we studied different drugs targeting a specific protein on Ewing sarcoma, we found a compound called clofurobin and cladribine. These are FDA-approved drugs. There's a known mechanism that works in leukemias, but what we discovered was that instead of this pathway in Ewing sarcoma, there's an alternative pathway the drugs can work. And the purpose of the project is to make derivatives of this thing. So here's the molecule of this drug. By changing these arms and legs of the molecule, creating a different version so that it doesn't do this. It doesn't kill the normal cells, but only kills the Ewing cells through this alternative mechanism. So we're just making derivatives of a known drug to make it more specific for Ewing sarcoma. So I'll stop here, and just like Brian, I'm around during the coffee break and lunch break. We can chat, and you can ask any questions you have. Thank you very much.
It was just about the last slide. You mentioned repurposing existing drugs and changing a couple of bits. I just had one last thought, which was, is that then an open door, or is there just less approval process when you change something in an existing drug?
Well, so for that one, we have two options. Option one is true repurposing. Even without making any changes, that drug can be tried on Ewing sarcoma patients. Like, I'm trying to convince my clinician colleagues to try this. And their hesitation with this drug was they've been using this drug in treating kids with leukemia, and the side effects they've seen in those children, they were not sure if the patient with the Ewing sarcoma who would be eligible for this drug would have been relapsed a couple of times. So they've been heavily treated, and their bone marrow would be already depleted. And they were not quite sure if the patients would tolerate the drug. But it is still possible. I'm talking to different clinicians, so there's still possibility of testing the drug as is on children with Ewing sarcoma, but it's a decision the clinicians will make. When you go and make a different derivative of this, now it's a new drug. Because it's a derivative of an FDA-approved drug, it may be a little bit faster track because you know how much to give, you know what are the expected side effects, you know what to watch for. So it will be hopefully a faster track to get an FDA approval, but since it's a new structure, it will require a new FDA approval, unlike using an existing drug.
Thank you.
Great. Hi, thanks very much for a brilliant talk. I love your imagery of people changing coats. That really brings out some of the challenges, I think. So I'm with you completely about drug repurposing. We've talked about it, haven't we, together. There are a lot of drugs out there, not just that are being used in cancer patients, but are being used to treat other diseases. So actually, we don't have time to evaluate all these in preclinical, in early phase clinical trials. So I just wonder how you think we can best identify the drugs that are going to give the biggest hit for patients and accelerate those into the trials. What do you think we really need to address in our strategies, in our research strategies, to bring those to the early phase clinical trials, so we don't waste years testing lots of drugs in patients that aren't going to be effective?
So we are partially doing that too. I guess one way of getting a faster outcome is the initial screening that you do with small molecules. Instead of including every small molecule we can get our hands on, you can limit that library to only FDA and EMA approved drugs. So if you start with only approved drugs, and when you get your hits, it will be hopefully relatively quick transition to clinical application. And we had another project on rhabdomyosarcoma. On that project, the hit came out, we were about to publish that paper, is an old antipsychotic drug they were using for psychosis in the 60s, and they stopped using it, but it was also an FDA-approved drug. So if the follow-up experiments give the expected results, that will be a faster track to using an FDA-approved drug from psychiatry into cancer research. But the initial libraries, if you limit your libraries to already approved drugs, that will be the only way to accelerate it.
But what about how we evaluate them then?
Oh, I wish I had an answer for that. That's really a big, big, big challenge. And even then, even when we find everything works in the lab, we still cannot guarantee that it will work in the humans.
It's great to hear what you're doing, and it's great that you're here from the United States. And there's obviously some collaboration between kind of people like the Bone Cancer Research Trust and academic work. But how much is the fact that we've got so many different government bodies, for the FDA you said there, the equivalents in Britain, Europe, and throughout the world, would them working together more make a big difference to bridging that gap? Because there's a lot, obviously, effort going on throughout the world, but could we bring that effort together more and help bridge the gap you talked about?
But I wish they do, but now we're talking into different governments working together. You know, they cannot even work within their own government with each other. So definitely it's a good idea, definitely it should be done, but I don't know how to make them do it.
But it would make a difference.
Hi. Both yourself and Dr. Compton, you've highlighted the fact that, you know, we're obviously talking about rare diseases, and the barrier to entry is in part the pharmas because they can't see a relevant market. But I know you have, as with other illnesses, broken down that barrier to some extent, and some drugs get through. But there's another barrier to entry, which I think perhaps less so is the FDA, because in the USA, for example, you have a very different medical system, funded very differently to the UK. But in the UK, our barrier to entry to getting drugs to the coalface is often nice. You know, back to your point, the regulatory bodies, when they see a drug that the NHS potentially has to pay an awful lot of money for, as a result of there only being a very small market, my experience is that often NICE are very reluctant to give you a clear gateway to entry for access to the drug for patients. They'll make it the drug of last resort, or, you know, you must do all sorts of other things before you touch this drug because it's so expensive. How do we in the UK break down that barrier? Or don't we?
Again, I guess the easiest solution would be you would need to get more government support behind that.
I'm not sure it's the government, though, that drive that. I
If
think
somebody
it's
has to
often
pay
doctors.
for it, it will be probably easier to get the government to pay for it than individuals.
There is a big role for us as a charity when it comes to campaigning to try and influence, you know, adopting new drugs, and it's something that we've done in the past. I'm looking behind you to Kate, or wherever she is. You know, it's something that our charity's been involved in the past when it comes to mefemertide, for instance. You know, that there was big resistance to adopting that as a treatment option in this country, and BCRT got heavily involved in that process in the very early days to make sure that that actually was approved for patients, because there was a value for osteosarcoma patients. And the reality is, should new treatments become available and get to that stage, that's where we will come into force and really get behind it and have to campaign to try and influence the decision making. And like I mentioned earlier, we need to work with other charities as well to have a combined effort in that front. And I know from speaking to some of my counterparts, it's something that we're all aware of. And actually we all have this shared view that we will rally and campaign to get approval where we can moving forward when new drugs come to that point.
And another example in the US, the example Brian gave about the RACE Act, it's like the stick, the government forces drug companies to do something. And the carrot part of that is there's an incentive the government came up with. It's about to expire. I'm not sure if they're going to renew it or not. Normally, there's a long waiting time for any drug to be approved by FDA. If it's a hypertension or diabetes, doesn't matter. Companies wait months and years to get their drugs approved, even if everything is ready. So there's a new program in the US. The government gives these drug companies a voucher. They say, if you bring a drug specifically for pediatric cancer, we will give you this voucher, and it is a get in front of line card. So if you get a drug approved for pediatric cancer, you get it in front of line, now you don't have to wait 2 months, and you can use it for any drug. So if drug company is waiting for a hypertension drug, bringing it to market 6 months or a year ahead of time could mean $1 billion for them. So that's an incentive for them to make drugs for pediatric cancer. And also government allows them to sell this voucher to other drug companies. So there have been 2 or 3 examples so far, and they were sold for anywhere from $60 million to $350 million between companies. So you can create incentives for the drug companies to make drugs for rare tumors, and I guess that's how the free market will make it work.